Biology in Space by Deborah Shokwade, Kira Pugh, Cheryl Rountree, Isabel Walsh, and Ikram Osame. It has been recorded that astronauts experience dysregulation in various parts of their body, especially after long-duration space flights. This is mainly due to the increase in oxidative stress in space. Reactive oxygen species can attack and damage parts of the cells, such as the DNA, proteins, and membrane lipids, leading to damage. This has led to the search for countermeasures to combat these dysregulations.
Upon exposure to the space environment, Apollo lunar astronauts are at an increased risk of developing cardiovascular disease due to the increase in reactive oxygen species in the body. It was found that lunar astronauts showed a higher cardiovascular disease mortality rate caused by heart failure, myocardial infarction, or stroke in comparison to their counterparts who experienced the same space environment at low Earth orbit only and did not experience space travel. The two factors that contribute to this increased risk are radiation and microgravity. With regards to radiation, the exposure to deep space radiation like hazardous radiation from galactic cosmic rays and solar particle events are believed to be the cause of higher cardiovascular risk to these astronauts, causing events such as ischemic heart disease and stroke. Microgravity refers to the weak force of gravity in the space environment. Like radiation, microgravity induces and upregulates the formation of reactive oxygen species and as it causes an increase in superoxidative levels in the brain cortex and in other regions of the body. Radiation and microgravity have been shown to have a synergistic effect on each other, leading to reactive oxidative species production and lipid perioxidation. Radiation and microgravity have been shown to have a synergistic effect on each other to give rise to reactive oxygen species production and lipid perioxidation, both of which lead to an elevated risk of cardiovascular disease.
Unwanted exposure to radiation of normal tissues, and especially radiosensitive organs, such as the lungs, can occur during space travel, where crew members may be exposed to galactic cosmic radiation and protons from solar particle events. Radiation exposure is identified as a potential health hazard to crew members during space travel. However, the effects of radiation on the pulmonary system are largely unexplored. Specifically, unrecognized galactic cosmic radiation-induced lung damage leads to imbalances in lung sphingolipid signaling and reduced anti-apoptotic and proproliferative levels of sphingosine 1 phosphate, and also an increase in tissue senescence markers. More noticeably, radiation exposure can cause chronic breathing problems in crew members, such as a cough and decreased exercise capacity, and pulmonary conditions and lung fibrosis, which can be fatal. A major candidate for protection against radiation injury is secoisolary serestanol diglucoside, or its novel synthetic LGM-2605, which scavenges free radicals and triggers an antioxidant response. This protects cells from a senescent-like phenotype and therefore reduces oxidative stress in human lung cells of crew members.
Microgravity and radiation increase the levels of reactive oxidative species, which affect the liver by altering protein and lipid metabolism, leading to tissue damage. This was proven by a study done on mice that was sent to space for 30 days, which showed decrease in bile transport proteins, which lead to accumulation of bile in the liver, causing tissue damage, as well as increase of fatty acid binding proteins, which initiate adipose cell differentiation, result in non-alcoholic fatty liver disease, and upregulation of PBAR alpha pathways, indicating impaired beta oxidation, leading to lipotoxicity and non-alcoholic fatty liver disease. These results, together with the downregulation on NF-kappa B, indicate decrease in oxidative defense, leading to inflammation.
So, in summary, the space environment provides many challenges to the human physiology, with examples such as its effects on the cardiovascular, respiratory, and immune systems shown and described throughout this video. All of these emphasize the impact and relevance of unstable superoxide production and continuous oxidative stress and damage that happens during spaceflight. So it is necessary to find forms of countermeasures to put in place in order to reduce these health risks for further space exploration, especially with the planned mission to Mars approaching in the near future. This could be carried out by professionally selecting radio-resistant people for spaceflight, supplementing free radical scavengers to astronauts, such as caffeine or glutathione, to stop the damaging effects of superoxide species, or cytoprotective agents such as amifostine to stop superoxide production altogether. However, with all these solutions being beneficial in the short term, if a form of Armageddon occurred and civilization had to inhabit a foreign planet, a long-term solution to these damaging effects would be necessary. This may be solved through gene therapy, with one such example being expression of radioprotective transgenes. Many organisms already exhibit remarkable degrees of radioresistance, and if these genes and molecular mechanisms can be elucidated and translated into humans via gene therapy, then this may constitute an effective strategy for enhancing radioresistance in humans for deep space exploration, thereby stopping effects of oxidative stress and damage caused by the radiation environment of space. Thanks for listening.